During translation within the cell, the sequence of nucleotides in the mRNA is translated into a sequence of amino acids in a polypeptide or protein. Cells decode mRNA by reading their nucleotides in groups of 3 called codons. In May of 1961, scientist Marshall Nirenberg, with the help of his post-doctoral fellow Heinrich Matthaei, deciphered the first 3 of the triplet codons in the genetic code. At this point in time, no one knew which nucleotides on mRNA were involved in creating which specific amino acids. Nirenberg decided he wanted to decipher the entire genetic code. They decided to use a radioactive approach to isolate every single amino acid. They first created a mixture of cytoplasm, ribosomes, and mRNA to create a protein. They would then add 19 non-radioactive amino acids and 1 radioactive amino acid. This way, they could track which amino acids were being coded by which strand of RNA being put in. They observed to see if the radioactive amino acid was incorporated into the protein. They did this 20 times with a different single amino acid being radioactive each run. They did this process first with a string of polyUs, meaning a strand of many uracils, and the only time that they got a radioactive label incorporated was with phenylalanine. In other words, the only time they formed a protein was when the radioactive amino acid was phenylalanine. Using this same method, they discovered that polyA codes for the amino acid lysine and polyC codes for proline. They could not find what polyG coded for, but they were able to discover the first 3 of all 64 codons. In 1964, Nirenberg continued his experiments with a different scientist this time, Philip Letter. They used a slightly different procedure this time. Short mRNAs with all different codon combinations, not just homopolymers, were added to a mix of ribosomes and tRNAs attached to amino acids. They still had 1 radioactive amino acid and 19 non-radioactive amino acids in order to determine which amino acid would bind to the ribosome. This was put through a filter, and if the tRNA bound to the ribosome and codon, it was caught by the filter paper, and all the tRNAs that did not bind fell through. The sample was tested for radioactivity, and if it was radioactive, that meant that the trinucleotide coded for the radioactive amino acid. Using this method, they were able to discover the remaining 61 amino acid and codon pairings. They were able to crack the entire genetic code. They were also able to identify that AUG is the start codon, meaning this is where translation will start. Translation continues until a stop codon appears. The 3 stop codons are UAA, UAG, and UGA. These act as stop signals indicating that the polypeptide is complete, where it will then be released from the ribosome. With only a few minor exceptions, all living organisms on Earth use the same genetic code. Because of the hard work of Nirenberg and many other scientists, we were able to decipher the entire genetic code, a remarkable accomplishment that benefits us still today.